Before I introduce the speaker today, I just want to first of all acknowledge that we're meeting today on the land of the Wurundjeri people from the Kulin Nation, and I'd like to pay my respects to them, their elders past, present and emerging. So it's my pleasure today to introduce our speaker, Dr. David Persing. David is the Executive Vice President and Chief Scientific Officer at Cepheid. He spent most of his 33-year career in biomarker discovery, translational medicine, and innovation in the diagnostic space. So Dave joined Cepheid in 2005, where he developed an industry-leading portfolio of products focused on the establishment of molecular diagnostic technology to meet global challenges in infectious diseases and oncology. He conducted his scientific and medical training with Don Ganem and Nobel laureate Harold Varmus at the University of California, San Francisco, and obtained his MD and PhD degrees in 1998. After residency training in laboratory medicine at Yale, he had leadership roles in academia and industry starting in the early 1990s, with the design, implementation, and scale-up of the first PCR reference laboratory at the Mayo Clinic. His interest in the democratization of molecular diagnostic methods has been long-standing, starting in 1993 with his publication of the first 5 widely adopted textbooks to include PCR protocols and guidelines for laboratory operations. He's published over 300 peer-reviewed articles and reviews, including multiple high-impact publications in journals such as the New England Journal of Medicine, Science, and PNAS. In 2020, he was named the Fierce Pharma list of the 22 most influential scientists in the fight against COVID-19. And to maintain his connection with the latest trends in translational medicine, Dave also serves as a consulting professor of pathology at Stanford University School of Medicine. So with that, please join me in inviting Dave to the stage. Thank you, Dave.
So thank you. It's a pleasure to be here. I was actually here 10 years ago. 6 months after the Doherty Institute opened. And this was after we had launched our MTB RIF test, which was one of the first tests to go global in its impact. And a lot of excitement about the fact that this technology could evolve to the point where it could actually be brought out to where it was most needed. Versus being locked up in a reference lab, which is where I grew up. I grew up in reference labs and then appreciated the need for really helping to push the technology out to where it needed to be. And one of the main points I'll make today is this is sort of a bit of a PCR history lesson. I've grown up with PCR and gone through the growing pains of the technology as it evolved, and give you some examples of what is happening with it, where it's going. And then one of the final points that I'll make is that, NGS right now is sort of where PCR was not 40 years ago, but maybe 20 years ago. NGS is the technology in need of democratization. Technology needs to be closer to patients and faster in turnaround time and delivering more actionable results. And I think that's really, it's the evolution of technology that needs to be recognized to really, needs to evolve to the point where it has maximal impact. So I'm going to, so past, present, future. So I'm going to talk a bit about the past before I talk about the future, and then we'll get into the present. I just finished hiking the first 2/3 of the Camino de Santiago in France and Spain. And so I was slightly underemployed during that period of time, but the best thing about being slightly underemployed is that you have time to think and think about the past, the present, and the future. But I think the most important point about this quote from Watson was that his discoveries were as a function of collaboration, and that's really the case in my career as well. Is that many of the key, most important applications of our technology come out of collaboration, whether it's developing a test or whether it's implementing a test and to create maximal impact. And a lot of those collaborations have happened with folks here in Australia. So I wanted to highlight some of those points. You know, there's a list. Everybody has their own sort of favorites list of what are the top innovations in molecular biology, and but you know, PCR is on the list, and it was actually invented over 40 years ago, 1983. And the message of this is, in even technologies like CRISPR, you know, we think about it as a new technique, but actually it was really Cas9 gene editing goes back to 2012, and we're just now seeing some of the practical applications of that technology. So it takes time for technology to make it out into the world. This was an instrument that we had at UC San Francisco when I was a graduate student. It was the only thermocycler in the university. This is a prototype gene expert, prototype Perkin-Elmer thermocycler. Didn't do detection because we didn't have real-time PCR at that point, but just did thermocycling. And the amazing thing was that you didn't have to add enzyme after the end of each cycle because you didn't kill the enzyme because it was thermostable. So being able to do continuous cycling with a thermostable enzyme was a major development. And we signed up for this instrument at all hours of the day and night for, you know, 1 or 2 hour, 3 hour blocks to be able to do our experiments. It was a long waiting list for being able to use this instrument. But in 1987, when I was doing tissue culture in the laboratory, I heard a radio report. Radio was playing, National Public Radio, talking about this new technique called polymerase chain reaction and its potential implications in diagnostics in particular. And this sounded really interesting to me. And so I sort of made a decision. Within about a month, I looked into it a lot. I talked to friends, talked to people I knew at Roche, which owned PCR at that time. And I had decided that I was going to pursue this as a major focus of my pathology and laboratory medicine career. So this was a pivotal moment for me to be able to understand where my career was going. And the reality of it is that PCR itself didn't become a mainstream laboratory technique until the development of real-time, self-contained real-time PCR with amplification and detection in a closed system. Up until then, we had amplification, and we had to take the amplicon out of the tube and analyze it, usually on a southern blot, with radioactive probes or later chemiluminescent probes. And this was a hazardous procedure because when you took the amplification product out of the tube, you subjected the laboratory to contamination. And this exposed our personnel, our laboratory spaces, the benchtops, the hoods, and the lab coats, the skin, the hair of our laboratory technologists became contaminated. So a whole lot of effort went into controlling the beast. And we had to create special labs. I was charged with building out the first PCR reference lab at the Mayo Clinic, and that required bringing in architects to design the facility and build it out. And so I went to Roche Molecular Systems in Alameda, California, where they were developing the first generation PCR tests to learn how they controlled the beast, and got their architects involved and ultimately built out this facility at the Mayo Clinic. So incredibly powerful, but this artist's depiction of PCR was a bit prophetic, I think, in showing that the entire world fills up with amplified DNA if you're not careful. Any one of those copies that in the orange horizon could cause a false positive if it got back into your samples. So we were, and there were a number of articles being published in the early 90s about contamination, potential for contamination. And so I had jokingly thought that my lab techs would all have to look like Sinead O'Connor, because of the potential for lab staff contamination. So we spent a lot of time on learning how to control the beast. One of the techniques we used in the lab routinely was sorrelin cross-linking of amplified targets. So we put sorrelins in a reaction to begin with, did the reaction, and then after the reaction was done, we exposed the tubes to UV light, and that cross-linked the DNA inside of the tube, rendered it inactive from an amplification perspective. But because we use isosorrelins, we didn't cross-link both strands of the DNA. We just cross-linked one strand. So we could still open it up and hybridize it and carry out analysis. Couldn't sequence it, but we were able to do hybridization. So we published a lot on this. And part of my job for democratizing the technology was to publish our protocols and our recipes and put them in textbooks. And we showed that this technique was very effective at killing essentially the amplicon from being reamplified. So we could still hybridize it as shown on the right. We saw a sorrelin and UV dose-dependent effect on the molecular weight of the amplicon based on cross-linking of the sorrelin. We also compared it against another technology called UNG to UTP, which incorporated DUTP in the reaction mixture, and that was recognized by an enzyme called uracil N-glycosylase, which chewed up the amplicon based on the DUTP incorporation. We also showed that neither protocol, either sorrelin cross-linking or UNG, was effective for short amplicons, especially if they were GC rich and didn't have many AT cross-linking or insertion locations. So we still had to be careful with, especially with short amplicons. So we published these and got a lot of citations for this work in terms of getting the technology established so it could be usable in a reference lab setting. Then along the way, we got a phone call. So what do all these agencies and acronyms have in common? They were all affected by amplicon contamination. And the OJ part stands for OJ Simpson, right? So the white Bronco, right? That's OJ driving the white Bronco, trying to escape the scene of the crime, right? So I'm walking in the hallway at the Mayo Clinic. I get a page. We didn't have cell phones back then. Pick up the phone in the hallway, and it's one of the members of the defense team for OJ Simpson. And they were trying to, based on what they thought was a contamination problem in the forensics lab, they wanted my opinion about whether it was a contamination problem, number one, and whether they could exclude the DNA evidence in the trial on that basis. And so I agreed to review the data. I expected maybe a file folder of information, but I got stacks and stacks and stacks of documents to go through for this, and it was a lot of work. But ultimately, Barry Scheck, who's a well-known forensic attorney who actually runs the Innocence Project in New York, was the guy behind this. And so I had to review all the data, and I decided not to go on the stand because I felt like they were going to try to taint all the forensic evidence in this trial on the basis of just the DNA problems they had with contamination, but which they did. They used that successfully to insert doubt into all the forensic evidence, and I didn't feel that was right. I thought all the other forensic evidence was quite valid and usable, but was discounted in its value. But anyway, life went on. 1994 was when I got that call. About 1994, we published data on an interesting project that we undertook 2 years earlier, and that was to see if we could actually detect tuberculosis and drug-resistant tuberculosis, at least one marker in one step. If we could do it directly from a patient specimen. So the idea was we had to go after and find out what was in the RPOB region, beta subunit of RNA polymerase for rifampin resistance. We could find a unique signature that we could use to amplify TB specifically out of clinical specimens. And so we used E. coli primers. The only sequence for RPOB at that time was in E. coli. We used E. coli primers to try to amplify stuff out of microbacterial DNA. And sure enough, we got a few hits. We got some amplicons that showed up on the gel and sequenced them, and then used those same primers against other mycobacterial species and related organisms, and showed that you could actually come up with a unique signature that was found within M. tuberculosis that was different from all the other organisms that we were targeting. So we made primers and made an assay. We made a hemi-nested assay that would be able to amplify this sequence directly from clinical specimens. We published on the ability to pull it out of directly from clinical specimens and predict rifampin resistance. But my PhD grad student at the time was, or postdoc, sorry, was wanted to put in the manuscript the potential for this technology to actually, make a difference someday in managing tuberculosis patients. And I was not too optimistic at that time that this lab technique, which was super, centralized and required DNA sequencing, would ever get out to the rest of the world. But nonetheless, I let him, wax poetic about the possibility of that someday. And then, sure enough, it happened eventually with the development of the MTB-RIF assay. But in the meantime, be careful of what you publish, because the Mayo Clinic staff would call and ask me to do the dreaded TB test, which I hated doing because it required a big effort, a bunch of staff, contamination control. It was nested PCR, which is still very much contamination prone. We had a dedicated facility. We had an army of technicians to run this. It took about a week to do everything and get a sequencing result. And if we had to charge for it, it would have been thousands. The cost would have been hundreds. So really not possible to see where this is going to go outside of a reference lab. But we use it in cases like this. We had a patient who flew into the Mayo Clinic from Saudi Arabia, member of the Saudi royal family. He had a 20-year history of treatment for pulmonary tuberculosis, poorly compliant with both TB drugs and his diabetes medication. As we know now, diabetes is like one of the worst things you can have to go along with tuberculosis, because the bacterial loads are very high, very hard to treat in diabetic patients. And so he developed a drug-resistant strain of tuberculosis. And he wasn't high-ranking enough in the royal family to merit his own private jet to Rochester, Minnesota. So he flew on Northwest Airlines, now Delta, first class, of course, London to Minneapolis, and he exposed people along the way. On the flight, he arrives at the Mayo Clinic with really nothing in the way of medical records to even indicate that he had a problem. And when the infectious disease staff found out that he had a history of drug-resistant tuberculosis, they all covered their, and brought masks in, and kind of a, scramble to manage the case. Exposed many people before this was found out. Follow-up study was ordered, and we actually looked at all the people who were potentially exposed to this person, And we did induced sputum studies to follow up on everybody who was exposed. So yes, they volunteered to do sputum induction, which is not trivial. I don't know if any of you have ever had that happen to you, or seen it happen to anybody else. It's not pleasant. But they did it. And we found that one of the X-ray techs had consistent detection in a blinded analysis of M. tuberculosis DNA over multiple weeks' time in these collected specimens. So we're concerned that when we amplified it and sequenced it, it was drug susceptible, not drug resistant. So he got exposed to TB from somewhere else, not from this person, but was exposed in a prior event. And he never PPD converted. The only way we knew that it was a confirmed case was that a guinea pig, and we actually used a guinea pig to look for evidence of infection. None of the cultures were negative. All the cultures were negative. No culture came up positive. Only PCR, and a guinea pig was positive. So confirmed case. But that says, hey, tuberculosis is a spectrum of infection. It's not just straightforward. You have it or you don't. It's actually, we'll talk about later, it's a spectrum of asymptomatic individuals who are carrying it and potentially infectious, along with people who have encountered it and resolved it and become latent TB cases. But it's a spectrum between latent and active and everything in between. As it would happen, this person on the plane from London to Minneapolis sat next to an HIV positive person. So, unfortunate consequence, or unfortunate coincidence. So anyway, the technology itself was established in the 1990s, but the practical impact was very limited. This really was dependent on having an integrated system that could process a sample, actually carry out the PCR process, and generate results in closer to real time to really have full impact. So democratizing PCR is what needed to happen. And my kids have informed me that the OG stands for the original gangsta. And that's what the expert was when it came to diagnosis of tuberculosis. So I became a consultant at Cepheid in 2001 and saw the evolution of this technology. And in 2001, it was something else that happened. Anybody know what this is about? Anthrax. Yeah. These envelopes were filled with anthrax spores and sent to the Senate office building. 11 people died. 5 people within the postal system, within the mail distribution center that sent these envelopes to the White House, to the Senate office building, died. And so this created a huge problem, huge issue for the country. They were very concerned about how to deal with this. Is this going to keep happening? Is it something that we can, can we even control this? So what they decided to do eventually was to implement PCR screening of the mail on a large scale. This was really the first demonstration of decentralized PCR testing in a low-skill environment using an automated system. So we worked with Northrop Grumman, which developed the mail sorting equipment, to develop this system, a collection hood that would collect air over envelopes as they were being sorted into different bins for distribution by zip code, and a cabinet that had the PCR detection capability built in, robotics, air concentration system in water, and allowing large numbers of envelopes to be tested with one cartridge. And they showed that that in a very controlled environment, they showed that that we could detect between 30 and 50 anthrax spores would be sufficient to detect deadly doses of anthrax in a single envelope among 10,000 envelopes to be able to actually implement this program. So didn't have to run one cartridge per envelope, right? You sniff 10,000 envelopes worth of air and you concentrate it and you test that, and that's enough to ensure the safety of the mail system. So this was the largest biothreat detection program. It's still in place in the US. We're still running this test every day, every hour in mail sorting facilities throughout the country. This is what it looks like in the post office. Mail comes in, it gets put into these systems that read the zip codes and then sort the envelopes into different bins. And over the curve in the line where the envelopes get pinched a little bit and air gets expressed out of the envelope, that's where the action happens. That's where the spores fly out. And so the collection system would collect spores, bring it into the system, concentrate the material, and then it would be tested in a single cartridge every 30 minutes, every hour, but the assay time was 30 minutes. On top of the box was this bar, different colored lights, right? Green, yellow, and red. Red means a hit, a positive. 2 virulence factors from Bacillus anthracis detected simultaneously in one reaction. The green was all clear, good to go. Send the mail off to the respective zip codes in the bins as sorted. The yellow light was might be a hit, might be something to look at. So and fortunately, the red light has never gone off. Green light's gone off a lot, like, every day, all day. But about a few hundred times per year, they will see the yellow light light up. And they need to investigate. They actually have done culture work to show that the aqueous concentrate from these lines, you can recover bacillus organisms from these concentrates. And these bacillus organisms may have one of the 2 virulence factors present. So the yellow light meant was a partial hit, one of 2 targets. We didn't have a 2-target system, we'd have a lot of false positives, and we would not, that would shut down the mail system within facility. So this resulted in a big infrastructure for decentralized testing, data monitoring in real time. And this was really, and over 190 billion pieces of mail have been tested so far in the system because of that 1 to 10,000 ratio. And presumptive negatives have happened, but they have not proven to be true Bacillus anthracis. But this, most importantly, this is the ability to show that you can actually do this method, PCR, in a low-skill environment and do it reproducibly and get data that's believable from this system. So fast forward from Mayo Clinic in 1994 to 2005, we started working on this test, supported by the Gates Foundation for development of a direct from specimen test for tuberculosis and drug resistance. And big study, big project, 5-year project from start to finish. Ended up with a publication in the New England Journal that had a big impact and resulted in WHO recommendations, or actually to endorse the technology for implementation. And this was really the OG for TB testing, right? For direct from specimen TB testing. We've produced about 130 million cartridges to date for this program. And it got a lot of coverage, a lot of press. The machine that will help NTB, that was the title of an article in MIT Technology Review. And they talked about a woman who got diagnosed quickly because, and she was concerned because she has chest pain. Her brother had been recently diagnosed with MDR-TB, and she was concerned she had it. But unlike her brother, who took months of time to figure out that he had MDR-TB, she got a gene expert test and she had a diagnosis the next day. And so this was, an example of what can be done with this technology. In the article, they commented that the machine resembles a high-end espresso maker. So I couldn't let this go. I had to take this a little further to help illustrate the difference between what molecular testing was like before this on-demand technology versus now. So coffee maker analogy, can I take your order, please? And I'd like a double decaf latte with a twist of lemon. I'll put your order in right away. It'll be available to pick up next Wednesday. Why so long? Well, we run our lattes in batches 3 times a week, so you just missed the cutoff. And that was the way molecular testing was done, even in Africa, where they had molecular testing. It was done in batches, it was centralized, and you wait for a long, long time to get the results back. And many of the patients never got their results because of that long wait, and loss to follow-up is a huge problem. So, and this is actually a sputum specimen. It kind of looks like a latte from the side. I know I don't groan, but this was actually a really challenging specimen to work with in the lab. It was viscous because of the cell-free DNA in the sample, made it really viscous, and it had to be inactivated for it to be useful in a decentralized testing setting. So we had to do all these things, and we figured out a way finally to do it after about a year of toiling with this. We used sodium hydroxide and isopropyl alcohols in combination to help solubilize the sputum and make it filterable so we could concentrate on the filter in the cartridge. And many studies had happened showing the impact of the point of care technology, showing that expert microscopy were able to deliver results basically the same day, whereas molecular testing sent to a central lab often never got to the patient, even after 140 days, but even then took a long time to get results back. So many of the patients with drug-resistant tuberculosis would have died before they actually got the drug resistance result. So this was viewed as a game changer, and it really had a big impact. So it was not without its challenges, though, and hasn't been without its challenges. We had to, in order to be able to supply this test in the volumes that were required, we had to get a subsidy from Gates Foundation, Unitate, and USAID to buy down the price from $17 down to $10. And that consortium actually happened. It made it possible for us to roll this out at a reasonable cost for countries to bring it in. But installation of systems in remote locations and training is challenging. Maintenance can be challenging in remote locations, especially in dusty environments where when you have a system that depends on optics to read fluorescence, and if dust gets in the system, it's challenging. So contamination problems are infrequent, but they do happen. We've seen some labs that run this test, have been running this test in culture rooms where TB culture is done. And there are TB genomes, flying around, and they have to move it to a clean location to get negative results. And then even at a $10 price point, the price was a barrier. We then reduced the price to around $7, but it's still a barrier in some settings. So how do you get around some of these challenges? We have been working with Beijing Chest Hospital to actually sort of make the most out of a single cartridge using a pooled testing strategy. And what they've been interested in is using the technology to do active case finding in patients who otherwise would not get tested. These are often asymptomatic or subsymptomatic or patients who don't feel that sick, not sick enough to get tested. And they've noticed that over a 10-year period that the number of patients without symptoms of tuberculosis based on their own studies had gone up from 12% to 43%, probably based on the fact that they were getting better at recognizing active cases, and that there were a lot of cases they were not otherwise going to be testing. And so they are really interested in addressing this large pool of asymptomatic cases. And even among smear positives, they found that 26% of the patients were asymptomatic. And this seemed to be a bigger problem in the elderly, where for some reason they were just not as likely to have symptoms that were driving a clinic visit. So, and how do you do this? Well, you can't really expect to induce sputum in somebody who doesn't have symptoms. They're not going to comply. They're really not, it's going to be hard to do that. So come up with different specimen types that actually can be used, that are easier to collect, the patient's not going to object to collecting. So deep throat saliva, clearing the throat, getting a little phlegm in the saliva, has been a technique that has been explored, and it works. It's not as good as BAL, but we've shown that with the Ultra test, which is a more sensitive version of the TB test, that about almost 80% of patients who are positive by culture in BAL samples are positive from deep throat saliva collected specimens. So it's not bad, not terrible. But they wanted to take it a step further, and they said, well, gee, that test cost is still too high for us to apply this to large populations. So how about if we pool samples in ratios of either 5 samples to 1 test up to 16 samples for 1 test, and test the pool. If the frequency of infection is low enough, we won't have to break apart every pool. We'll just test a lot of patients and screen them as negative, and then break apart only the positive pools to find out which one is positive. And this, they started this on around 17,000 people in a pilot study. It was published. They screened out patients who didn't have positive chest X-ray findings. They later decided they didn't want to use chest X-ray for as a rule-out tool. So, but they had 1,800 individuals who were recruited for the study for pool testing, and they had 346 pools that were negative and 4 pools that were positive. And they had 4 patients that later were confirmed. Out of those 4 pools, they were confirmed to have active infection. And when they looked at the patients, they actually saw that a number of them were asymptomatic and had negative chest X-rays. So now they are in the process of scaling this up after this pilot study to over 30,000 individuals, and they're not using chest X-ray as an exclusion, abnormal chest X-ray. So they're scaling it up in a way that they think will lead to identifying more asymptomatic carriers of TB and better population level TB control. And there are other reasons to pursue different specimen types for tuberculosis diagnosis. In fact, latent tuberculosis is very common. Roughly 1/4 to 1/3 of the world's population is going to test positive for latent tuberculosis, and those patients can reactivate tuberculosis during their lifetimes. And this shows the sort of the spectrum of infection, and subclinical TB, which we just talked about, is part of it. Latent TB needs to be addressed as well. And so why do we want to detect it? Well, because of the potential for reactivation, and those reactivated cases become infectious. And we need to know who to treat prophylactically with latent tuberculosis to prevent reactivation. And that's been a goal. So, and there's really no tests that can distinguish between active tuberculosis and latent tuberculosis in the same test. So we've been working on this is the test that everybody uses, the interferon gamma release test, uses one biomarker, gamma interferon, to measure gamma expression after incubation with TB peptides. And they use 3 or 4 tubes per patient to be able to do this. And it's a lot of work to do this in the lab. So we're working on a one blood tube assay that we think could have an impact. And we're actually focusing on being able to distinguish between active and latent tuberculosis by looking at the expression profile of a bunch of markers. Certainly interferon gamma needs to be in there, but a bunch of chemokines and other markers are, we think, going to be able to help us distinguish between active and latent by looking at the ratios of expression of these different mRNAs after stimulation by TB peptides. So it's a quantitative multiplex assay that will look at gene expression profiling as a means of identifying cases and potentially stratifying them as well between active and latent categories. So early results look quite promising. And this is one of the projects that we're working on to come up with a different specimen type that can be used instead of sputum, especially if it works for active tuberculosis. So this is one of 3 projects that we're working on the host response side. I won't talk about the other 2, but the sepsis subtyping assay to distinguish patients who can benefit from steroids in sepsis versus those who might be hurt by steroid administration. This is a project with Oxford that we're collaborating on. And then a bacterial versus viral assay to enable antibiotic stewardship decisions in real time in patients who have fevers and who could be septic, but again, based on gene expression profiling. One of the projects that we worked on in Australia and really started from a collaboration here in Australia was the hepatitis C finger stick test that we worked on with the Kirby Institute. Jason Grubbely and I met at a meeting in 2015, and he saw the viral load test that we were working on and said, hey, that thing looks really interesting. It works on plasma, myeloplasma, but do you think it might work on a finger stick blood sample? And I said, well, I don't know. It's 100 microliters of blood max you might expect out of a finger stick. Half of that's plasma, so it's 1/20th of the volume that you're putting into the test to get a sensitive enough result out of that test. Is that going to be good enough? And so he ended up doing a lot of the initial work to show that if you put 100 microliters of blood from an HCV positive person into the assay, that would work. Actually had high concordance with a reference lab test. And then we converted the test to finger stick blood version, which works directly from finger stick blood and generates results in about 60 minutes or less. If you're positive, you actually have the benefit of having an earlier result call out because the cycling just stops when it hits the point of positivity. So this has led to some big programs and big studies that have been conducted here in Australia, and ultimately the TGA approval of the test and rollout of this technology into needle exchange clinics, treatment centers, and bringing the technology closer to the patient. And this cuts out a lot of the steps in terms of getting a result that could be treatment directing. So the idea is to test and treat in the same day, if you can, and avoid loss to follow-up with the current protocol, which requires antibody testing and then bring the patient back and get us another sample for RNA testing and then wait for the result. And loss to follow-up happens at every step. And it's led to really moving this technology all throughout the country on the Combi Clinic, Volkswagen van with HCV testing instrumentation in the back, going from place to place in prisons and treatment centers and screening programs. Pretty amazing what has happened in Australia with this technology. the studies have shown that this results in less loss to follow-up. And when you lose patients to follow-up, you've actually wasted testing capacity on them because they never get linked to care. So this difference in loss to follow-up is so significant that it does make sense to bring this kind of testing closer to the patients in hard to reach populations and hard to engage populations. This has been shown throughout Europe as well in multiple programs. And all these publications finally caught the attention of the US CDC. They said, hey, you know, we have a big opioid epidemic in the United States, and we have a lot of people who are injecting drugs because of that. And we have a lot of hepatitis C. It's actually growing, not going down. We need a point of care test solution. So this led to ultimately to the CDC, NIH, the White House, the Biden White House on developing a national elimination strategy for hepatitis C, led by in a bipartisan manner with Senators Van Hollen and Dr. Cassidy, that have led to the development of a point of care HCV test and release in the US and US FDA trials that were needed to do that. So it's exciting, but I have to say, you know, if it weren't for Australia, if it weren't for the Kirby Institute, if it weren't for Jason Grebely, if it weren't for the experts in implementation science that have really made the difference here, we would not be in this position in the US to have this test. So it's exciting. I'm going to move ahead. So really thanks to Australia for showing the US what good looks like. DNA sequencing. This is you know, I mentioned earlier that NGS is an area that needs democratization. It's located in central labs, requires a lot of skill, a lot of time, but it's kind of like PCR was decades ago, and it needs to move to more decentralized locations. And so our vision for this technology is to make it easier to use and on demand. And it's not there yet, but we can make parts of it easy to use and on demand. And some of the most labor-intensive steps of NGS are the sample prep and library prep steps that go into creating a library for each sample. So even with automated instrumentation, it takes hours of tech time and hours of wait time to make a library for NGS. So if we can make that much easier, much less labor intensive, and automate it, and do sample prep and library prep in the same system and get it ready for a sequencer, irrespective of the back end, whether it's Illumina or Oxford or PacBio or other sequencing platforms, would that be a way to at least introduce the technology to a better way of doing NGS? So this is probably especially relevant in the infectious disease space, where in contrast to oncology, infectious disease demands most of the applications, the best applications would require a faster turnaround time than 2 or 3 days, which is what it takes right now. So we've done some work actually to show that we can, inside of that cartridge, with the rotary valve in the base of the cartridge, we can access all those chambers. We can move things between chambers, move things into the PCR tube, incubate it at a constant temperature, pull it back, do some more things, add linkers, add primers, add reagents, and then push it back into the reaction. We can do all that with the rotary valve technology that enables, you know, manipulation and fluid movement inside of the cartridge. And so we've been able to show that we can go to from a 4-hour, 20-minute time per protocol with an hour and a half of hands-on time down to an hour and 54 minutes, even less with some protocols, and only about 5 minutes of hands-on time, and get the same depth of genomic coverage for a whole genome analysis of a drug-resistant chlamydia pneumoniae strain. So getting all these drug resistance genes, we showed this between the QIAGEN sample prep and the Cepheid library prep cartridge, that we actually got more even representation of NGS results across the bacterial genome with the automated approach in the cartridge. So someday, this may become the subject of democratization tool, and this is just a first step of being able to show the potential for this to happen. So we also recently announced a collaboration with the Fleming Center, which is focused on antimicrobial resistance, and they're super interested in faster ways of getting NGS data on their patient samples. They're interested in sepsis subtyping, steroid benefit versus do no harm. They're interested in bacterial versus viral infection discrimination from an antibiotic stewardship standpoint, trying to retain antibiotics for those who really need it. And they're also interested in identifying ways of figuring out who's a super shedder of a carbapenem-resistant organism. We know that patients come into our hospitals already colonized with carbapenem-resistant organisms due to prior healthcare exposure, and that's a major source of hospital transmission. Is the shedders, the people who are not in isolation, who are still nonetheless shedding carbapenem-resistant organisms, the carriers of these organisms in their GI tracts. So one interesting project is to look whether C. difficile plays a role in turning a patient from a sort of regular shedder into a super shedder, because if you get diarrhea, you can imagine this becomes more of a biohazard in a hospital setting, right? And control of both C. difficile and CRE. If they're co-colonized with CRE, this could be an opportunity for CRE to spread even more efficiently because of the patient condition. So, but right now, you know, we wait on even ordering a C. difficile test until the patient has had multiple diarrheic episodes. So I think we have what could be called a three-potty problem. We wait for 3 episodes before we even order a C. diff test, and that's an opportunity for contamination with both C. diff and CRE if they're co-colonized. We can also look at cycle threshold values of CTs, of CREs in the GI tract. Maybe with these factors and cofactors come up with a way of predicting who really, really, really needs to be in strict isolation versus who can be in just, you know, contact precautions. And so this may be a way of better addressing the problem, because their isolation resources are limited like they are everywhere in terms of who really needs strict isolation. But the major point I wanted to make is that that we depend heavily on collaboration or innovation. And it's a model for how we can leverage our relationships with academic centers. Our collaborators often come up with novel technologies that we haven't even thought, novel ways of applying the technology we haven't even thought of. And this is, for us, a huge opportunity and a great opportunity for patients to see this technology get to where it needs to be. Many of our innovation programs have occurred in Australia: hepatitis C with Jason Grebely, HPV with Andrew Vallely and Marion Saville, CTNG and group A strep with Rebecca Guy. These collaborations have really fueled a lot of the way we think about how our technology can be implemented, how we can maximize the impact of it. And this is a really important area for us to keep focusing on, especially for future collaborations, is to figure out ways to leverage this technology in the best way to make the maximum patient impact. So, past, future, present. And the present is, I'm done with my talk. So I'd like to open it up for questions or comments. Yeah.
Thank you, Dave, for such a brilliant talk, which is highly relevant to many of us here at the Doherty. And that one of those first slides that you put up of that old PCR machine really takes me back to my PhD days many years ago. So it's amazing how far we've come since then.
Yeah.
Any questions for Dave?
Thank you, David, for the excellent presentation. You talk about decentralization of testing. I'm very interested in your view, or with your view or Cepheid, in terms of the future, whether there's plan to make the technology more accessible. For example, the GeneXpert point of care molecular tests or the NGS platform that you develop, whether it's going to be an open platform for people to use, develop their own assay, put it on, and use the platform for detection or characterization of less prevalent pathogens or neglected pathogens, shall we say.
Yeah, that's a really good question. I get asked that a lot, as you might imagine. We have a couple of programs that actually do that, where we work with an academic center to develop a new test. We work with USAMRIID, which is the U.S. Army biothreat program that's administered by NIH. And they're developing tests at USAMRIID for exotic pathogens that they might be worried about in the future, like Nipah virus, for instance. So we've enabled some of these partners to actually figure out how to do test development on their system. David Land, who really takes credit for a big chunk of the innovation required to make the MTB RIF test, because of the use of molecular beacons to be able to scan that RPOB region for mutations. His work was done entirely within the university, and we coached him on how to be able to do that. So it's possible in the future we could actually do this more with more partners to be able to figure out ways to collaborate and allow this to happen locally.
I wanted to ask about whether Cepheid would potentially be looking at metagenomics and.
Can't quite hear you. Sorry, can you hold the mic up closer?
Yeah, of course. Thank you for the talk. I wanted to ask if you would be looking at metagenomics for diagnosis of infections.
Metagenomics. More complex libraries, perhaps, but probably similar to what we used for the Klebsiella project that we just showed. Yeah, I think it could be possible. I'm not sure the diversity of organisms we'd be able to cover, but we would need to, to see whether the existing approaches would be sufficient to cover, because we're using shotgun-based approaches for whole genome sequencing anyway, using random hexamers for priming. So potentially that could be applied to metagenomics.
Thank you. I really enjoyed the entire talk. I'm just interested in, when you assess what you might put your resources into, does it have to have a big global application? Are you, as Thomas said, looking at sometimes smaller pathogens or less common pathogens? How do you decide what Cepheid will invest in?
In terms of packaging efficiency or environmental impact of packaging? Yeah, was that the question?
Oh, I think it was just more about, are you focusing on big problems globally?
Yes.
Is that the priority for you? What is a big global problem?
It has been a historical focus of ours, is to respond to emerging demands. We were brought in to develop the first decentralized test for monkeypox, Mpox, which in the US got FDA clearance and CLIA waiver for testing in decentralized settings, and central labs for that matter. And so we have a history of when there's an urgent need for a new test, we are able to usually rise to the occasion and respond fairly quickly. Same for Ebola in 2014. I'm sure that will continue in some capacity.
For one more question, Peter, at the front.
Yeah.
Thank you. I am actually interested in the environmental impact of the cartridges themselves. That wasn't going to be my question.
Yeah.
I'm not a TB guy, but the test you're developing to look at gene signatures between latent and active, are those gene signatures likely to differ across different races of people? You know, Africans versus Asians versus Caucasians, for example?
Yeah, we have looked carefully at that to make sure that we have population diversity built into our discovery activities, and we're finding markers that are common among cross populations. So we're very concerned about that, and we want to make sure that the signature we would develop would be universally applicable.
And can I ask you to comment on the sustainability, environmental impact of cartridges and disposal of cartridges?
Yeah, it's really a big focus of ours right now to come up with ways of using less plastic to make the cartridges, using bioplastics to make the cartridges, using sustainable materials for packaging, coming up with better ways of cartridge disposal. I was just talking with Mary and Saville earlier today, and they were having the same problem with like vaccine, and during the vaccination, getting rid of all the used syringes and, you know, just the huge amount of medical waste that produced. So it's something that we in the industry need to be focusing on, is better ways to manage waste. Sure.
Well, thanks very much, Dave, for a wonderful presentation.
Thank you.
And please join me in thanking Dave for his